Hi, I'm Sarah.
Hi, I am Farah.
Hi, I'm Bashira.
Hi, I am Yusra, and in this video, our group will be present about ELISA.
ELISA. ELISA stands for Enzyme-linked Immunosorbent Assay. There are two basic principles of ELISA. The first one is the antigen is immobilized to a solid surface. Then, the antigen is complex to a detection antibody conjugated with a molecule amenable for detection, such as an enzyme or a fluorophore. ELISA assay is typically performed in a multi-well plate, either 96 or 384 wells, which provides the solid surface to immobilize the antigen. Immobilization of analytes facilitates the separation of the antigen from other components in the sample. This characteristic is what makes ELISA as one of the easiest assays to perform on multiple samples simultaneously. To understand better of the principle of ELISA, let's look at the basic setup of an ELISA assay. From the picture, we can see a capture antibody on a multi-well plate will immobilize the antigen of interest. The antigen will be recognized and bound by a detection antibody conjugated to biotin and streptavidin HRP.
Now, I'm going to present about levels in immunoassays for ELISA. First, what is immunoassays? Immunoassays are biochemical tests used to detect the presence or concentration of a specific molecule such as hormone or toxins in the solution of antibody-antigen reaction. So, what is antibody? Antibodies are Y-shaped protein produced by immune system of humans and other animals in response to foreign substances, which is antigens like bacteria, toxins, and viruses. Antibodies are antigen specific with the binding site of antibody antigen like key and dot. And with this bonding relationship of antibody antigen, it is used in immunoassays to detect a specific molecule. In immunoassays, the antigen is called as analytes. Now we are going to talk about which enzyme involved in ELISA tests. In this table, it shows the most two common enzymes that are used in ELISA tests. Firstly is HRP, which is horseradish peroxidase. Secondly is AP, which is alkaline phosphatase. Chromogenic substance is peptides that react with the protolytic enzyme under formation of color. They are made synthetically and are designed to possess a selectively similar to the natural substrate for the enzyme. The chromogenic substance for HRP are DAB and TMB. DAB is diaminobenzidine that used in membrane ELISA procedures as precipitating substrate for the detection of peroxidase activity. DAB is the substrate of choice because it produces an intense, easily observed brown stain. TMB is tetramethylbenzidine, which is soluble substrate yield a blue color when it detects HRP. The color changes to yellow with the addition of sulfuric acid or phosphoric acid with maximum absorbance at 450 nanometers. It is sensitive and may produce significant background signal if too much protein or antibody is used. TMB oxidizes quickly compared to other HRP substrates, resulting color development. So, for the chromogenic substance for AP are BCIP or NBT and PPNP. BCIP or NBT is used as set of chromogenic substrates for enzyme-labeled alkaline phosphatase. After catalyzed by AP, the product BCIP, which is 5-bromo-4-chloro-3 indolyl phosphate, it further reacts with NBT, which is nitro-blue tetrazolium, to produce an insoluble precipitate that is dark blue to purple in color. PPNP, which is p-nitrophenyl phosphate, it is substrate used to detect alkaline phosphatase in ELISA application. It is yellow water-soluble reaction product that absorbs light at 405 nanometers. It available in crystalline powder, 5 mg tablets, or as ready-to-use formulation. Lastly is chemoluminescent substance. It is a property of chemical reactions that emit light as a byproduct. The chemoluminescent substance for HRP is ELISA bright. ELISA bright is highly sensitive chemoluminescence for HRP. It has the highest signal-to-noise ratio for superior performance. The broad linear dynamic range, it can enhance detection and precision. The immediate light generation, it can cause fast detection. It is also cost-effective because it reduces reagent consumption. The chemoluminescent substance for AP is diazotine base. It is 1,2-diazotine compound. utilized in AP. It produces light signal when it is activated by alkaline phosphatase, accumulates in its dephosphorylated form, and decomposes at constant rate for up to several days. When assayed on nylon membrane at 1 to 100 in buffer provided, CDP star signal reach a maximum within 50 minutes and decay slowly over 3 days.
ELISA is classified into 4 major types. The first one is direct ELISA. In direct ELISA, only enzyme-level primary antibody is required, meaning that the secondary antibody is not needed. The enzyme-level primary antibody directly binds to the target antigen that is immobilized to the plate. Next, the enzyme linked to the primary antibody react with the substrate to produce a visible signal, which is producing a color change that can be measured. In this way, the antigen of interest is detected. Number 2 is indirect ELISA. In indirect ELISA, both the primary antibody and secondary antibody are required during the detection stage. But in this case, the primary antibody is not labeled with an enzyme. Instead, the secondary antibody is labeled with an enzyme. The antigen of interest is first coated to the plate. Then, the primary antibody is bind to the antigen immobilized to the plate. Following this, the enzyme-level secondary antibody will be introduced, and then it will bind to the primary antibody, which will in turn be bound to the antigen. Finally, the enzyme will link to the secondary antibody will react to its substrate to produce a visible signal that can be measured. Number 3 is sandwich ELISA. In direct and indirect ELISA, it is the antigen that is immobilized to the plate. In sandwich ELISA, however, it is the antibody that is immobilized to the plate, and this antibody called capture antibody. In addition to capture antibody, sandwich ELISA also involved in the use of detection antibody, which generally include the unlabeled primary detection antibody and enzyme-level secondary detection antibody. Firstly, the antigen of interest is bind to the capture antibody immobilized to the plate. Secondly, the primary detection antibody bind to the antigen. Thirdly, the enzyme-level secondary detection antibody will bind to the primary detection antibody, and then the enzyme will react with the substrate to produce a visible signal, often involve a color change, which is the release of light or fluorescence that can be measured. Number 4 is competitive ELISA. Compared with the 3 ELISA types before, the competitive or inhibition ELISA, also known as blocking ELISA, is perhaps the most complex of all the ELISA techniques. In fact, each of the 3 formats, direct, indirect, and sandwich, can be adapted to competitive format. The competitive ELISA is predominantly used to measure the concentration of an antigen or antibody in a sample by detecting interference in an expected signal output. Essentially, sample antigen or antibody competes with a reference for binding to a limited amount of labeled antibody or antigen respectively. The higher the sample of antigen concentration, the weaker the output signal, indicating that the signal output is inversely correlates with the amount of antigen in the sample.
I am going to present to you about example tests of ELISA. For first example is diagnosing infection such as HIV. Diagnosing infection is usually an antibody testing that done on blood sample. In this test, a person's serum is allowed to react with virus protein that have been produced in laboratory. If the person has been infected with the HIV, the antibodies in the serum will bind to the HIV protein, and the extent of this binding can be measured. The next example is pregnancy test. Pregnancy test is done by a mixture of purified HCG that linked to an enzyme, and the test examples are added to the test system. To get more understand, you may see the picture of test diagnosing infections HIV and also example of pregnancy test that use ELISA techniques.